First of all, I'd like to thank the organizers for inviting me to present my work on the interaction of eggs and sperm at this exchange session. And before delving into the mechanism of fertilization, I would really like to draw your attention to the different steps that we can highlight, and they are depicted here in panel B in this figure. First of all, the acrosome reaction, in which the content of the acrosomal vesicle in the sperm head is released. And this reaction is essential for the sperm to be able to pass through the zona pellucida. Despite the fact that this is an essential reaction, we still don't know whether the acrosome reaction occurs when the sperm contacts directly the zona pellucida or before that, and also we don't know exactly how the acrosome reaction is triggered. After that, only once the acrosome reaction has taken place, the sperm can pass through the zona pellucida, and once inside the perivitelian space, the sperm binds to the egg membrane. And really, I want to focus your attention on the two different steps here, the binding and fusion. So at the binding stage, the membrane of egg and sperm come into close contact, but it's only at the fusion step that the membrane merge together. And this is a very expensive event, energetically speaking. And usually for this reason, there is a protein known as fusogen that induces the fusion of the membrane of cells. And until today, there is no known fusogen in the acting in the during sperm-egg interaction. So we don't still know the identity of the fusogen on the sperm or on the oocyte. After fertilization, the mammalian oocyte establish a block to polyspermy, which means the incoming sperm cannot pass through the zona pellucida and cannot fuse to the membrane of the oocyte anymore. This is to ensure that only one sperm fertilizes one egg, and the block to polyspermy is established both at the level of the zona pellucida and of the egg membrane. So there are changes happening there in both environments. But my main interest is in the molecules, cell surface molecules that orchestrate this interaction between sperm and egg. And specifically, I'm going to focus today on the interaction of membranes of sperm and egg. The interaction is depicted here schematically, and also you can see two beautiful images taken with scanning electron microscopy, showing again the interaction of the sperm with the surface of the oocyte, which is covered in microvilli, and the engulfment of the head inside inside the oocyte during and after fusion. Despite having this very well descriptive mechanism here and these images, we really don't know the details, the molecular details of this interaction. And when I first joined the Sanger, there were only few molecules known to be essential for this interaction. And when I say essential, I mean that the proteins were assessed in a mouse or in an animal model. So at the time, it was CD9 that had been shown to be essential for fertilization, because knockout animals were infertile, female were infertile, and also there was a family of proteins known as GPI anchor proteins that were known to be required for fertilization, and their expression was known to be required in oocytes, because by treating oocytes with an enzyme that cleaves the anchor and therefore releases all GPI anchor proteins, or by knocking out a gene that is required for the synthesis of the GPI anchor, and again creating oocytes that do not have GPI anchor proteins on their surfaces, they were able to create sterile female. And on the other hand, there was only one protein known to be essential for fertilization in on the sperm, and this protein was named Izumo-1 after a marriage shrine by the Japanese group that discovered the protein. So we set out to try and identify the binding partner of Izumo-1, and when we started our work, we actually didn't know whether Izumo had a binding partner at all on the oocyte. So to try and answer this first question, we used a method that we routinely use in the lab, so we can express the extracellular domain of Izumo-1, which is a single pass membrane protein. So we can express this domain here in our cell culture system. We express this domain as a soluble recombinant protein with a detectable tag. And to see whether Izumo had the ability to bind the oocyte membrane, we incubated ZP3 eggs with our probe. And we were particularly happy to see that the probe was able to bind the surface of oocyte, because that showed us that not only the probe, so the protein that we were able to produce in the lab, was functional, but also that very likely Izumo-1 had a binding partner on the surface of oocyte. So we used that probe to try and identify the binding partner for Izumo-1. We used the probe in a large screening assay. So we used a cDNA library made from immature oocytes, transfected cells with this library, and performed a screening, incubating the transfected cells with our probe and checking for cells that were positive for the binding to our probe. And as I said, after quite a large screen, we were able to identify few cells that were able to bind to our Izumo-1 probe. And when we sequenced the single plasmid that, when transfected into the cells, was able to make them binding to Izumo-1, that plasmid turned out to encode for a gene that was called, at the time, folate receptor 4. The name was given to this protein because of its homology to other members of the folate receptor family. But as you can see in this alignment here between the protein sequences of all the components of the folate receptor family, the amino acids that are actually directly involved in the binding to folic acid, and which are highlighted here in red, are different in folate receptor 4. And also, when we assessed whether folate receptor 4 was able to bind folic acid in vitro, we couldn't detect any binding. So indicating to us that the protein was playing a different role compared to the other folate receptor. And based on that, and on the fact that it played a role in fertilization, we proposed to rename the protein after the Roman goddess of marriage and fertility, which is called Juno. And also, we thought that that name made a good match with its binding partner, Izumo-1, which, as I mentioned earlier, was also named after a marriage shrine in Japan. And here is a statue of the goddess Juno that you can find in Rome. So the protein was able to bind to Izumo-1, but then obviously we wanted to check the function of the protein in vivo and in vitro. Therefore, we used, first of all, an in vitro assay. We added an antibody against Juno in our IVF assay. And that assay showed us that the antibody was able to block fertilization in vitro. And to further confirm the role played by Juno in fertilization, we generated knockout mice that showed us that Juno was required for fertilization in female, because male mice lacking Juno are completely fertile. I have to say that both male and female are viable and healthy, but females are infertile. And when we checked exactly the reason why the female were infertile, we could confirm that the absence of Juno on the egg membrane induces the inability of the oocyte to be fertilized by sperm. We could never detect any fusion, and we could never detect any pronuclear stage embryo when we did IVF assays with Juno knockout eggs. So, because we have established that Juno was required for fertilization, we then wonder whether we could be able to distinguish between the two steps that I described at the beginning, binding and fusion. So first of all, we checked the ability of sperm to bind to Juno knockout eggs, and we saw that Izumo and Juno is definitely required for binding, because sperm were less able, or were able to bind less efficiently to Juno knockout eggs compared to their control. And then we wanted to check whether this binding pair Juno and Izumo was also sufficient to induce fusion in the cells. And to do so, we expressed the two proteins in two different batches of cells, and we mixed them together and checked for fusion events. But we could never detect any fusion between cells expressing Juno and Izumo, and this indicated that Juno and Izumo are required for fertilization. They are essential for binding, but they are not sufficient for fusion. And then we noticed a very interesting behavior of Juno, which is shed from the membrane of the oocyte soon after fertilization, and it is no longer detectable at the pronuclear stage here. You can see the staining is lost from the membrane. And when we checked with electron microscopy, we could see Juno in these dots here that are very likely vesicles, small vesicles, in the perivitellin space. And so we proposed the idea that maybe Juno could be involved in the establishment of the polyspermia block. I just wanted to remind you that the establishment occurs both with modifications at the zona pellucida, but also at the membrane level. So there is definitely a block at the oolemma in fertilized eggs. But very interestingly, the polyspermia block is not established when eggs are fertilized by ICSI. And so we reasoned that we could check the expression in ICSI fertilized eggs, and we were reassured to see that Juno was actually maintained on the surface of oocyte fertilized by ICSI. So you can see here the expression of Juno even at the pronuclear stage, while this doesn't happen when eggs are fertilized by standard IVF. So the model that we propose is that the binding of Juno and Izumo is essential for fertilization, and after fusion, the shedding of Juno from the egg membrane not only reduces the ability of sperm to bind to the oocyte, but probably the presence of this vesicle in the perivitellin space also actively blocks incoming sperm by acting as decoy eggs. So suggesting that really Juno is involved in the establishment of the polyspermia block. But so far, the data I have presented all refer to the mouse model, but recently it has also been shown that Juno is required for fertilization in human. It has been shown that the protein is definitely expressed on human oocytes, and also by using antibodies that bind Juno, it is possible to block fertilization in vitro using human gametes. And there is also one case reported of a woman with infertility that has a homozygous missense mutation in which an amino acid in Juno is modified. And so both data strongly suggest that Juno is required for fertilization in humans as well. As I said, when I started my work at the Sanger, there were very few proteins shown to be essential for fertilization, but last year actually other 4 proteins on the sperm had been shown to be required for fertilization, adding to the picture. One of these proteins is TMM95 that was actually identified a few years before in the cattle industry. So the group that identified the protein was able to narrow down homozygosity region in a subpopulation of bulls that were severely subfertile. This mutation was a nonsense mutation in the transmembrane region of a protein called TMM95. And as I said, the bulls were severely subfertile. Then a colleague in Spain generated the mice knockout for this very same protein, TMM95, and it was able to show that knocking out that gene induces the same phenotype as Izumo1, meaning that when you do fertilization in vitro, you can find sperm inside the perivitelline space, but fusion never occurs between sperm and egg, and so fertilization is not achieved in vitro or in vivo. So clearly the next question that we tried to answer was whether TMM95 directly interacted with Juno and Izumo. And when we tested that in vitro, we didn't see any interaction between the two of them, while we have the positive control of the interaction with Izumo and Juno, and the reverse orientation is also true. But when we added TMM95, we never saw any interaction. And so we then asked whether TMM95 could possibly be the fusogen protein that we were looking for. And to test that, we used a slightly different approach to the one used previously. In this case, we transfected TMM95 in, again, 2 cell population, but this time these 2 cell population express Izumo, Juno, and split GFP. In this assay to detect fusion, you basically have the 2 halves of the GFP in 2 different populations, and it's only when fusion occurs and therefore the 2 halves of the GFP are in the same cytoplasm that protein is reconstituted and fluorescence can be detected. So we had a positive control, which is syncytin here, which is a well-known fusogen expressed in the placenta, and you can clearly see the green signal due to fusion taking place into between the 2 cell population, but we never saw any green signal when we transfected in TMM95. And this indicated that TMM95, like Juno and Izumo, is required for fertilization, but is not sufficient to fusion. And I have to say, it's not entirely clear whether TMM95 has a binding partner on the oocyte as well. So the picture has changed over the last few years, as I was mentioning. The other protein other than TMM95 that have been shown to be essential for fertilization are SOF1 and SPACA6 and FIMP. For all of them, it's yet unclear whether they have a binding partner on the oocyte and whether there is an interaction, direct or indirect, between all of them. So there are still many questions open in the field. Possibly the most pressing one is this identity and existence of a fusogen, but also there are other questions that would hopefully be answered and investigated and answered over the next years. And with this, I'd like to conclude. Thank you for your attention. Thank my PI at Sanger and people I work with at the Institute, our collaborator in space, Maria and Pablo, and the 2 funding bodies, the MRC and the BRCC.